Today, we're diving into the complex yet exciting world of genomics data interpretation, specifically through Genome-Wide Association Studies, or GWAS.
That's fantastic. I've been eager to understand more about GWAS and how bioinformatics plays a role. Where do we begin?
Well, let's start with the basics. GWAS is all about assessing genetic variants across the genomes of many individuals to see if any variants are associated with a specific trait. It's
Right.
become standard practice for studying complex diseases.
But with so much data, isn't there a risk of false positives? How do researchers handle that?
Exactly, that's a major concern.
Yeah,
Because GWAS involves millions of statistical tests, multiple testing corrections are essential to minimize false positives.
so they use methods like the Bonferroni correction or false discovery rate adjustments?
Yes, precisely.
Got it.
Bonferroni is conservative, dividing the desired significance level by the number of tests, but it can be too strict and increase false negatives.
And the false discovery rate is a bit more lenient?
Exactly. It controls the expected proportion of false positives among the declared significant results. It's about finding a balance. But here's the kicker. Because of linkage disequilibrium, or LD, SNPs aren't independent.
Oh, that's when genetic variants are correlated, right?
Yes. So, instead of adjusting for millions of SNPs, they use a standard threshold of p less than 5 times 10 to the negative eighth. It's become the
Interesting.
convention in GWAS.
That makes sense. But what about population stratification? Doesn't that affect GWAS results too?
Absolutely, it can introduce confounding if not properly accounted for.
Right.
Researchers use methods like principal component analysis or mixed models to adjust for it.
So, they include principal components as covariates in their models?
Exactly. This helps control for ancestry differences within the study population.
Gotcha.
It's crucial, especially in admixed populations.
But even within the same self-identified race, there can be genetic differences, right?
Yes, and that's why meticulous adjustment is
Yeah.
key. Failure to account for population stratification can lead to inflated type I error rates.
Okay, so moving on, what about phenotypic heterogeneity? How does that impact GWAS?
Great question. Phenotypic heterogeneity refers to the variation in how a disease presents among individuals. It can drastically reduce statistical power if not properly addressed.
So, defining the phenotype precisely is critical?
Exactly. A well-defined and consistent phenotype ensures that the genetic associations found are meaningful.
Right.
Otherwise, you might need a much larger sample size to detect associations.
It's like trying to hit a moving target if the phenotype isn't consistent.
Precisely. And researchers also need to establish that the trait is heritable before conducting GWAS.
Of
No
course.
point in looking for genetic associations if genetics doesn't contribute to the trait variance.
That makes sense. But I've heard about the issue of missing heritability. What's that about?
Ah, yes. Even when a trait is heritable, GWAS often doesn't account for all the genetic variants. This missing heritability could be due to variants with very small effects or rare variants not captured by standard SNP arrays.
So, how do researchers tackle rare variants in GWAS?
They use different statistical methods. For common variants, individual SNP analysis works, but for rare variants, they aggregate them. Yes,
Like
exactly.
burden tests? But burden tests have limitations, don't they?
They do?
Yeah.
They assume all variants have effects in the same direction and magnitude, which isn't always true. Methods like SCAT, or Sequence Kernel Association Test, address this by allowing for different effect sizes and directions.
So, SCAT can detect associations even when variants have opposing effects?
Exactly. It's more flexible.
Understood.
But it's less powerful when all variants affect the trait in the same direction. That's why methods like SCAT-O combine both approaches.
It's all about finding the right tool for the data.
Absolutely. And after identifying significant variants, researchers often perform pathway analysis to make biological sense of the results.
Okay.
It helps in understanding how different genes interact in biological processes.
So they map identified genes to known pathways and see which ones are enriched?
Yes, that's the idea. Tools like gene ontology and CADD are commonly used for this purpose.
But aren't there challenges with pathway analysis in genomics?
Definitely. One challenge is that SNP-level data need to be mapped to genes, and the methods for doing this are still evolving. Also,
Interesting.
pathways are dynamic and context-dependent.
Right, so a pathway might be active in one tissue but not another.
Exactly. So researchers have to interpret results cautiously.
Yeah.
Integrating other omics data can provide a more comprehensive picture.
Speaking of which, how do polygenic risk scores fit into all this?
Great point. Polygenic risk scores, or PRS, aggregate the effects of many genetic variants to predict an individual's risk of developing a trait.
Got it.
It's particularly useful for traits influenced by many genes with small effects.
But they require careful validation, right?
Absolutely. They need to be tested in independent cohorts to ensure they predict accurately and aren't overfitted.
Right.
Cross-validation helps assess their performance.
And what about the ethical considerations, especially with diverse populations?
That's a critical issue. Most GWS have been conducted in populations of European descent. This limits the applicability of findings to other ethnic groups.
So increasing diversity in genetic studies is essential?
Exactly. It improves the generalizability of results and helps reduce health disparities.
Absolutely.
Plus, allele frequencies can differ across populations, affecting disease risk.
It's like trying to use the same key for different locks. It just doesn't work.
Well put. And finally, replication is crucial in GWAS to confirm findings.
Right.
Due to the risk of false positives, validating results in independent cohorts strengthens the evidence.
And functional validation in model systems adds another layer of confirmation.
Exactly. It bridges the gap between statistical associations and biological mechanisms. It's all about building a solid understanding of how genetics influences disease.
This has been incredibly enlightening. It's clear that interpreting genomics data requires careful consideration at every step.
Absolutely. From study design to analysis and interpretation, bioinformatics plays a pivotal role. It's an evolving
Indeed.
field, and staying updated with best practices is essential.
Well, I think we've covered a lot of ground today. Any final thoughts?
Just that collaboration between clinicians, researchers, and bioinformaticians is key to advancing
Agreed.
precision medicine. By working together, we can unlock the full potential of genomics in healthcare.
Couldn't have said it better myself. Thanks for this great discussion.
Thank you, and thanks to our listeners for joining us on the Hey Gen podcast. Until next time.